Thanks for coming to this CRB seminar. It's a real pleasure for me to be able to introduce our speaker today, Neil Kelleher. So Neil is the Walter and Mary E. Glass Professor of Molecular Biosciences, also a professor of chemistry, and has a number of other appointments here at Northwestern. He's the director of the Chemistry of Life Processes Institute and director of Northwestern Proteomics. We're going to hear a fair amount today, I think, about proteomics. This research group focuses on this area that, again, we'll hear about, top-down proteomics, and he's the founder of the Human Proteoform Project, a global research initiative that its aim is to weigh every protein in the human body. That is 250,000 proteoforms in 4,000 different cell types. This project will enable dramatic increases in the speed and efficiency in which investigators can identify higher value protein-based markers of disease and spur game-changing advances in biomedical research. Neil is a Fulbright Fellow. He received his PhD from Cornell. He completed a postdoc at Harvard Medical School. He served on the faculty at the University of Illinois State before joining Northwestern in 2010. He's received a lot of honors. The Arthur P. Sloan Foundation Fellowship at Packard, the Dreyfus Award for New Investigators, the Camille Dreyfus Teacher Scholar Award, a Lilly Analytical Chemistry Award, National Institutes of Health Career Translation Awards, and others. And he's also the co-founder of a number, I think 4 successful startups, as well as a software suite called ProSite that's used in over 1,000 labs around the world. So a very, very impressive set of accomplishments. And doing work now associated with the Proteoform project that it might be really exciting to those of us in robotics and automation. So I'm really excited to welcome you here today.
Yeah, thank you. Thanks. A fantastic introduction. Just, Ed gets up and walks in a snowstorm and delivers a fantastic intro. Thank you for the invitation. Thanks for the chance to, go to in a space that's on the periphery of what you do normally. And I very much appreciate that. So questions throughout, and my main goal is to do information transfer and see if there's not some major center institute level synergies that the, Northwestern could benefit from. And that means both sides needs to benefit strategically. So let me just dive into the wacky wild world of proteomics. That's, protein level biology. The inspiration is no less than the Human Genome Project level accomplishment, but to do it for human proteins, the products of our 20,300 human genes. So the scalability of that is an engineering ethos that is required to do the project. As much as basic scientists, chemists, biologists, others like to think about the biology drivers, right? And we need to answer that distinctly. And I will attempt to do that today with 3 major drivers. But that's really, in my career, I've been driven by that engineering ethos. It's otherwise known in the chemistry world as analytical chemistry, measurement science. That's what drives me. And so I've been at the intersection with biologists for this entire time and the field of chemical biology. And I come to you from the institute there, Silverman Hall, right, on the little lake next to the bigger lake, the little pond, that's Chemistry of Life Processes Institute. I took over that institute about 2 years ago and charged by then VPR, Milan Mrksic, who came to my first board meeting and said, we need to have a vision, a vision that would put Northwestern in a differentiated lane relative to anything else in the world. And it is difficult to achieve that, but this is what you're going to see, is there is such a lane, and it's at Northwestern in Chicago, Illinois. It's not in Massachusetts or California. And that's a little bit of strategy for you, the third coast. But what are we talking about at the molecular level? So it turns out the concept of a protein, right? If I have 20,300 genes in the human genome, and I say, oh, each one encodes a protein. A lot of molecular biologists say that, but it's imprecise. It's actually not true. Yeah, it's a protein, I get it. But actually what drives our biology are all the proteoforms, the distinct molecular entities that form through reversible modifications sometimes enzymatically driven, sometimes environmentally driven. You can cleave the protein, covalent bonds get cleaved, creating different isoforms. And I'm part of a community that then said, all of these like phosphorylation, acetylation, methylation, glycosylation, those are all different enzymatic processes that regulate, potentiate the function of these protein products from in our bodies. And it goes on all the time. It goes on every day. And so there is a proteoform program of life that we have yet to develop measurement tools to match the scale of our biology with the scale of our tools. We did it for the genome, at least at the DNA level, right? We were able to develop tools to not to first match the scale of our biology with the scale of our engineered solutions, and then go beyond. Now, genomics is an overdetermined science. By that, I mean you have 40x coverage. When you sequence your genome of your cancer, every base, you have 40 times redundancy measurement of that base. That is an A or a G or a C or a T. It's an overdetermined science, and there's an entire ecosystem, tens of billions of dollars a year in that marketplace, right? This has not been the case in proteomics. And so it's time to do that type of scale of things. And, you know, as part of a community, so I'm the founding president of this consortium for top-down proteomics. They came up with this word. It's not just a word and a molecular entity, it's a movement. And we have now 500 members of this. We're still yet the minority in the field of proteomics that is calling loudly for this project. But the chorus and the crescendo is rising. And it's just based on, here, let me slow down. And so from a single gene in the genome, you already have complexities that form. So you might have a gene, you would look at it in a computer and you'd say, oh, it's 500 amino acids long, right? String of amino acids for a protein. Here's two length variants that have a different, these are cartooned protein structures. And that has a different length than that. It's a bit longer, right? And this happens in a lot of different tissues from the same gene due to RNA splicing or at the intermediate RNA level. And then here come the decorations. So here come all the enzymatic, most of them reversible. So there's a logic that has formed for homeostasis and to keep us healthy and goes awry in disease. And this is just a single proteoform. So what we do at the chemical level, molecular level, is catalog these. We can determine in a process I'll give you some sense about the molecular weight, so weigh every proteoform. That's the Human Proteoform Project. And we can assign the position of these decorations, the total length variant with complete molecular specificity. That's the difference. That's top-down proteomics. The world's leading platforms for proteomics today, they use little antibodies to look at little pieces of the proteins, or they use little peptides. They'll digest this into pieces, and then it's like Humpty Dumpty, you know, all the king's horses and all the king's men. You can't put it back together again.
Is that bottom up?
That's bottom up proteomics, and that is the dominant, that's about a $5 billion a year marketplace and ecosystem, which is great and built a tremendous amount of proteomics knowledge in the world. And so I'm not trying to cannibalize that marketplace. I'm trying to build another one that's about as big right on top. Yeah.
What's coding SMP?
That is a single nucleotide polymorphism or a mutation. If it's present at 1% or above in the human population, it's a polymorphism. If it's more rare of a spelling difference, that is coding. So it's in the coding sequence of the gene, then it's a polymorphism. And so you often get these driving mutations in cancer. So we capture every atom in this molecule, including
Things like glycolysis,
glycosylation.
glycosylation. Do those just happen naturally based on like the form of the protein? Or is there something in
Yeah,
code?
yeah, like on your cells in your body, of which you have trillions of about 4,000, 5,000 different types of cells. On the surface of those proteins on the surface often get like this little sugary, it's a sugary coating that helps it be stable and long-lived on the surface of cells, for example. Yeah, and there's about 250 different types of post-translational modifications in biology. There's a lot of them, but the main action is right around about 20, 30 of them. Yeah, and you have like logic in your body, like here's a phosphorylation, for example, and that comes from ATP binding an enzyme will transfer the phospho group to a serine or threonine or tyrosine amino acid. And then there's phosphatases that take it off. So they're reversible. And you can put a couple of those phosphorylations on and the enzyme goes from inactive to active. Right? So this is the world that has evolved over 600 million years of evolution on the planet. Ever since snowball Earth thawed and we had the Precambrian explosion of eukaryotic life, this is what evolved, right? This is our biology. And it's just surprisingly difficult to get the world to confront that we have to sequence these molecules and we have to know the players on the field. Otherwise, drug development is going to be inefficient, regenerative medicine, you know, and all the rest. So we're talking about a transformation in our ability to know ourselves at the protein level. And it's because, you know, and so in this consortium, we have all sorts of use and success cases. But we are still the minority in the field. And it's going to take a technology and, you know, automation, scaling of technology to realize, to measure the fact that our proteins are not specific enough and they have all these different decorations. Like, you know, like an example where this is going on in tau, in Alzheimer's disease. Tau, we already can see over there in the Proteomics Center, you know, there's 600 different proteoforms of tau at the 1% threshold of abundance and higher. We can see it right away in an Alzheimer's brain relative to a normal brain. And we're tracking all, that's an example project. And we're doing that with the Feinberg School of Medicine. So measure proteoform signatures. And many of my excellent colleagues doing bottom-up proteomics, they say, yeah, well, you know, it would be great if we could do it that way, but we can't do it that way. We can't do it top down to get better proteoform markers. And they just go about their lives, and which is very successful group of scientists, but it's unlike cosmology or precision astronomy, where in that field, I'm sorry, but your next big telescope or experiment is probably $5 or $10 billion. And so your relationship to each other as a community is very different. Your relationships to governments and other stakeholders who fund you, very different. This is, you know, this is biology, chemistry. We're used to small ball and individual-led research, except for things like the Genome Project or the Brain Project, et cetera. So picking up some speed here. Protein isoforms and these things called post-translational modifications, those are the decorations. In our world, we call them PTMs. And this is going to be the, if the technology is in place, the most efficient way to find the function of protein isoforms and their modifications. If you measure things correctly in the top-down fashion, you will correlate to disease much tighter. So your ability to detect disease earlier will be much better. And that we have use and success cases built around that. And then this thing, it'll transform the drug industry. When I say I have a drug target and I say it's tau, the protein tau, what are you talking about? There's 600 different drug targets. What families of proteoforms do you need to target, right? That is an example of the kinds of impacts I mean, there's little question that that'll be true. The only question is which decade this century we're going to get there. And so here's some very granular bits out of my world. We use mass spectrometry because currently the discovery of proteoforms has to use mass spectrometry. There are new methods coming and some startups in this space, but for now, this is an example of two types of proteins, a histone and something in a signaling cascade called MEK1. These two proteins, these are the proteoform landscapes. This is what happens when you treat them with drugs, the anti-cancer drugs in cell line or mouse models. So these are the kinds of things that we can do now, but we're far cry from the scale of our biology. And so this is the end of the intro, but just to revisit, this is the expansion of proteoforms. There's sort of a proteoform explosion that also makes people a little fearful about approaching this area. What most people believe is this, in a single cell type, say you got cancer cells in a dish, how many different proteoforms might evolve? People have very different answers to that question. And the bottom-up community is developed tools that are so good that capture so many of these that community basically thinks this to be unwieldy, an unknowable scale of a challenge. But those of us in the top-down community go, yeah, no, we could see them. Sure, we'd like to get tenfold deeper and better, but no, it's not infinite. It's not. But if you do math and you say, oh, I have 40 of these modifications, and each one is a binary, 2 to the n, it's 2 to the n math, so really simple. 2 to the 40, that's a pretty big number of proteoforms. But in the evolution of our biology, it has created a program to constrict that explosion. This is the point that is not generally agreed to in the field, creating a lack of consensus, and that this is 2 to the 40 for this protein, not like 100 or 1,000 proteoforms. They think it's 2 to the 40. And it creates problems getting consensus in a field of leaderships.
But you're telling us that in fact, it's more like a 100 or 1,000 different forms.
Yeah, like tau, you can go 2 to the 90, 2 to the 100. But I can show you the proteoform map, at least down to say 5% relative abundance or 1%. You know, what's below there, it's an iceberg. But yeah, and people, but yeah.
So thinking about tau, are you saying that if you have 60 different proteoforms of tau, that some of them might be impacting Alzheimer's more than others?
Yes, absolutely, yes. That is already known. There is a phosphorylation on position 217 in tau that everybody's super excited about. But it's just one of those little balls. And they finally found, after 15 years of that phosphorylation, not in the brain, but in the blood, you can actually generate a test. And that correlates to your degree of tangles in your brain at some level. At least that's where we're at, 2025. Everyone's excited. And I'm like, but yeah, there's like 5, 6, 8 other phosphorylations. So how does that, you know? There you go. Right, so the scale is the last intro slide, and yeah. So to accomplish the project I will pitch to you, which is scaled to be 10 years and somewhere north of $3 billion for the deep version. There's a shallow version, so it's on sale. You're ready to buy it. This is the scale of the challenge, is that the cost and speed and the depth. So we need, over current technologies, we need to improve about 100-fold for both of those, cost and speed. And then the depth, yeah, this is a complicated discussion that we're going to skim over today, but you get a sense of, this is not a 10 to the 6 problem. This is about where the Genome Project stood in the early 90s. And that, the leap ahead is provocative. So the project is to determine, quantify the diverse proteoforms in the 4,000 cell types that Ed mentioned, for the 20,300 human genes, and oh yes, in the 12 body fluids as well. And to develop the technologies in 2 steps. One would be proteoform discovery, and then there's the equivalent of what is next generation sequencing, next gen proteomics, which is single molecule proteoform sequencing. But to get to that point, you really got to do the project right once first. And that's the equivalent of the Genome Project. And so I'll skip the 3 points for, you know, new technologies, other than to say there is aligning interest, both private foundations and public funding. And the technology sector around this is growing, is poised to embrace this. And the state of play in the technology space is interesting, and I do hope we'll have this thawing and the project moving forward soon. Right, so then I just have to the focal points, and here we can have a back and forth as well. What does a measurement scientist do to try and build technologies that can do this? We have a mass spectrometer involved. There's a lot of what we call the front end problem, and we bring just super basic commercial robots and lab automation to bear. That's all we got over here. Proteomics generally is under engineered. And so we grab a commercial robot for examples that I'll show you. I do have a startup in this space, so financial conflict of interest, but it is on a robotic system that's, you know, they sell 10,000 of those a year, very basic, for sample cleanup and then injection into the mass spectrometer. And then on the far right of the screen, you have this profile. These are actually high resolution, different antibodies that arose in someone to fight COVID. It's their antibody response to fight COVID, revealed at proteoform resolution. That kind of spectrum wasn't even possible 5 years ago. So let's go through the daisy chain, what we do. So we take some blood or plasma or serum, we drop it in this robot. It uses antibodies to affinity enrich a certain protein. And of course, all its proteoforms. And it's a very simple system. And certainly, if we were in a pharmaceutical company, there is a next level of automation that's possible around this. But you know, we're an academic shop and with those trappings. But this is what we do. It uses little the antibodies are on magnetic beads, and it uses a magnet to isolate them and then work downstream. Yeah.
Do you find that like some proteoforms that they have a lot of tags on them that like when an antibody for like that general protein won't bind to certain proteoforms?
It's a great question. Yeah, it's known that effect is very known, where if you have the decorations on it, it blocks. It's called epitope masking. But yes, absolutely. And it's an unknown. So sometimes we use 2 antibodies, like for tau, we'll use 2 antibodies to pull it, to enrich it and get it, so that we're not less subject to that. Then the question is, once you get that tau sample, how do you sequence it? How do you analyze it? How do you measure the proteoforms? So what we do then is release from the antibodies, from the beads, we release the tau, in this case, the target, from the bait. And I can explain this robot, et cetera, in detail, but basically what it does is drop the sample in there. And these are all postdocs and grad students and staff members, humans. This is what does not scale well. This is what gets tired. This is why I'm here. What options might we have to scale what we do? You know, when you bring it from the one semi-automated process to the next step, and no daisy chain in between, other than, you know, that with 2 legs and a heartbeat. And so inside this channel, in a process called sample stream that Phil Compton started this company 8-9 years ago, left my lab. It's basically a protein, you know, so here's the tau in red. It's a protein washing machine and concentration device. So you're dealing with nanograms of protein. You're dealing with very small amounts of protein. And so it comes, and then you get this cross flow force. It's a 10 microliter channel inside this, and held in like a little credit card device, credit card holder. And it'll concentrate it because of the different flows. And it's got a membrane, molecular weight cutoff membrane, to wash away the salts and soaps and get it ready to inject in the mass spectrometer. And because you're engineers, I thought I'd give you, there's a lot of ion physics and advanced mass spectrometry This is where I got trained, quite some time ago. And now we're just partnering with Thermo Fisher Scientific. It's very seldom that you'll find an academic building their own mass specs. I mean, yeah, their engineering, their R&E team is just, crushing it.
Could you walk us through those sort of the basics of how this works?
Oh, sure, yeah, so after, you deposit from the enrichment into these, trays, you clean it up. The sample now, a few microliters will make its way into the source of a mass spectrometer, which is a sort of electro sneeze. It's, this thing. So this is held at 3 kilovolt. And the fluid comes through, and in a Nobel Prize in 2002, there's an electro spray that forms. And it's not a super robust process. This is actually a fail point for too many experiments. So this whole situation needs a makeover, under-engineered. But once the droplets form, they're little micron droplets that then go into a vacuum system. So this is progressively, there's a little hole, so it goes from atmosphere down to 10 to the minus 11 torr through a series of stages of differential pumping through viscous and then molecular flow regimes, if you know those terms. And then what you get is this ion beam. So you get this beam of proteoforms that are now with no solvent. They're all dry, and now they're ions. And I forgot to mention, it's at pH 3, so it's acid, or lightly acid. And what that does is takes all the protons from the acid and they attach to the protein. So you have these positively charged particles, and they are multiply charged. So that's what's happening inside this. So on axis, you get this beam, and then this is called an orbitrap. It's taken over Fourier transform. It's the Porsche of mass spectrometers. Basically, it's high resolution. And I think do I have a video? I don't, of the different stages. But this is how you weigh every protein. We can weigh the ions that are formed and calculate a molecular mass. It's a mass spectrometer. And in the next step, I'll explain this too. When you get that ion beam, you can collect, say, 1 million or 10 million ions over here, and then take them out behind the woodshed, controllably fragment them. You collide them with gas or photons or electrons. It's all very fun. But it fragments the molecules into what is a fingerprint. And that's how we tell where the decorations are on the proteoform. Right, and so there's this super fun, 5 years ago, we got a $2 million grant, internal grant from Thermo, very competitive within the company. And my buddy from Cornell, who I got my PhD with, he's now the head of R&E. So hang in there, your network becomes valuable. So this fellow, Mike Senko, and we had this idea, and within a year and a half, we've cracked it, it works. I'm all excited to tell you about it, but I'm time limited. Basically, what happened is we went from an ensemble of ions to a single ion. Each single ion, we can now measure the molecular mass of each single molecule in the instrument. And it dropped the requirements, the sample requirements by 1,000-fold less concentration. This is a concentration sensitive detector. So instead of 1 nanomolar, 1 micromolar, you need 1 nanomolar concentration. This is propelled, and even if tau has 600 proteoforms, we can see them all because of this new approach. And it has to do with the constructive interference as the genesis of signal here in the old school way of doing it. And then each ion is its own genesis of its own signal. So it really is a change. And here's probably a good video. It's 40 seconds. It shows 4 of these particles. And there's the 4 being detected over about a second period inside this high vacuum, ultra high vacuum. And a lot of different procedures, but this is heavily robust. These are the four masses of the four particles spinning around inside there at about 50 megahertz to lower than a megahertz. That's the spectral range of the motion. Okay, and so we got into COVID like many people, driven by COVID research. We thought, geez, we can see all these proteoforms now. And this is another example. Let me come back. So the human immune system, you probably have this idea, or maybe even you remember from your classes that, okay, I have these thing called antibodies. There's a subset called IgGs. And those IgGs are in response after a few weeks. When you get sick, you develop new antibodies that can bind the pathogen. That's the molecular warfare going on when you get COVID. And they'll say, oh, well, the computational space of antibody production is such that you can create 10 to the 11 different IgGs in your body at any given time. Oh, well, that would just be way too many to measure, right? Yeah, not so much. I mean, if you sub-select, you go through the process I went through. There's two parts to the antibody, heavy chain and light chain. These are the proteoform patterns from the heavy and light chain components of all the IgGs that recognize the COVID virus. And this is just so we're the first group to be able to like, well, yeah, those are all the light chain, there's your lambda and your kappa, different classes of light chain, and these are all the proteoforms. And if you're really sick, you generate a lot of a pattern that looks like you were vaccinated. And if you're not really sick, this is automated. Wow, cool. We're getting into automation. I love it. So you guys are already helping us be more efficient. Yeah, this is a particular driven by aging and cardiovascular health. We want to get to, for example, 1,000 people. And we want to engineer a system to be able to get to, actually, I want to do 5,000 people. And that means I need 10 microliters of blood from 5,000 people of different ages and cardiometabolic health, et cetera. And this is the version. We're on version 2 now. We're going to version 3 to get these proteoform patterns from the blood. So yeah, it's a kind of project where you should see what a group of chemists over there are trying to do. I mean, I do have a lot of computer programmers and lab, we have real-time data acquisition. And so we can talk to the mass spectrometer and make it do different things. And I've got a really good group of about 8 programmers that are in that space. And yes, that's an offer to come meet and play with those guys and gals. Yeah, so I just got 2 little vignettes, and then we can chop it up in any way you guys want. We've used this sensitivity to do 2 things. And the first I'll just show you is proteoform imaging. So we have another system, automated system to move up just a glass slide with a thin section piece of cancer tissue on it. Okay, it's a 10 micron thick piece of human tissue, and it's going to be moving at 40 micron a second. And as it moves, we're going to sample it with this now ultra-sensitive proteoform detection technique. And so it's a little liquid junction there that takes it up to the electro spray source, and it moves it across that. So we can get images across the tissue at about a 40 micron spatial resolution. So spatial proteomics, it was the method of the year in 2024, spatial proteomics. So now we are the group bringing proteoforms into that conversation of spatial biology. Yeah, here's the kind of setup. It's not super refined, but that little liquid junction is sitting here. There's the plate, and we got it on a little automated system that is visually inspected and has non-idealities associated with it. Yeah, and so we get quantitative information out of this. I think, yeah, here's the bottom line. You get, so here's that piece of cancer tissue. A pathologist will say, okay, you've got definite tumor sections, and then you've got the stroma next to it. This is non-cancerous. And so I'm gonna show you just one protein that comes in 3 flavors. And it's because there's a decoration. So here's a protein sequence, N terminus to C terminus. And these are all the amino acids. And this arginine can be unmodified, monomethylated, or dimethylated at this arginine. That's 3. Then actually there's other decorations, but that's the one that's important for here. And so this is to the biology claims that how much does proteoform information matter, right? In this case, if you're unmethylated, you're really localized to what we now believe are smooth muscle cells around this artery. So there's a blood vessel right there. And that unmethylated form, at that arginine here, the naked form, resides in space around that artery. But then if you get mono and then especially dimethylated, you're more in the tumor sections of this. You're upregulated in the tumor sections of this clinical specimen. So that's an example. We have 2 or 3 others like this that, yeah, oh my gosh, I'm really interested in the enzyme that adds those methyl groups because that seems like it's upregulated in cancer and could be a drug target.
Is cause and effect established in this process?
The biology, like the control over the localization, like why would those cells around the artery be not methylating to then have whatever program they're running run? The amount of that is a 5-year 2 postdoc piece of molecular biology to prove and that's why I use caveats.
Yeah, sure.
Because you've got to find the methyl transferases, you have to knock them down, you have to, and then, it.
Yeah.
So, but the molecular discovery is solid. And yeah, the causality of it, and the utility of it, that's harder to say something hard. Yeah.
So this methylation comes from the tumor cells, or you modified the amino acid to have the methylation group?
The mainline statement would be in the tumor cells, which populate, there's thousands of tumor cells in this section. Each one of those cells, this growing tumor, right? Bad. You have an upregulation of what I believe an arginine methyltransferase enzyme that, for whatever reason, in the cancer program that's got out of control, that is part of that program. So you've just sensed the biology of that program.
Yeah.
Yeah.
But theoretically, if we modify the amino acid by terminating with methylation group,
Yes,
all amino acid will give the same information.
all amino
Because
acids.
it's hard to identify amino acids because of the same structure. There is not much difference.
Oh, I forgot to mention, that's it. Okay, that's a great question. Here, let me go back. So this is a proteoform in my world. We know if there's one proton out of place. The molecular weight measurement is 7 significant figures. It's ridiculously accurate. I forgot to mention. Right, you're walking across 1 Dalton space on the proteoform. So when I say there's a methylation, what I'm saying, yeah, so unmethylated, you add 14 Daltons, 14 grams per mole, 14 atomic mass units. You add every time you put on a methyl. So for us, that's a big shift. And that's why we see it. And that's why this is so freaking hard, I suppose, or why we haven't done it yet to date. And when I said at the outset, complete molecular specificity, yeah, I meant it. Like every atom on the protein. Yeah.
Mass spectroscopy will let you know what's been added, but not where. Where comes from your other work with the.
Yeah, you remember the woodshed comment?
woodshed,
Well, I've
yeah.
said many things in 40 minutes, so I don't expect you kind of to, and this is not there won't be a quiz. But what I meant behind, out behind the woodshed, when I was, talking about this guy. This picture, where'd it go? Yeah, right. Yeah, here. So this is out behind the woodshed. And what we do is we break covalent bonds. And in this case, it was with colliding it with gas. And what happens is, yeah, so all these blue flags, those are the pieces. So if I take a single molecule, single proteoform ion, and I start heating it up vibrationally, I'm gonna break a covalent bond at one position. It's gonna give me the N-terminal half and the C-terminal half, or the piece. So this piece is something like that ion, like this would represent this, and this would represent the other part, had we seen it. And so and then we have, 10 to the 5 ions that undergo this process. And that's how we, so formally, what we know is we have these different these 14 Dalton differences are between this region.
Yeah.
So it could be these, but that doesn't make any biochemical sense. You could methylate, a phenylalanine, but yeah.
Okay.
So I'm almost done. So we use the same basic approach of this liquid junction, and all we do is put a set of single cells. So the other thing the cool kids in proteomics school are doing these days is single cell proteomics. That's like the North Star. And as spatial and single cell proteomics have lifted off, I just felt like, oh man, the proteoform, what about the proteoforms people? And it just didn't compete, say 5-6 years ago for what would be the next big thing. But the voice you've heard today is not just my own, and it's rising in its volume. So single cell, all we do is on that slide, instead of putting a piece of cancer tissue, we actually create a slurry of single cells. So we take the tissue and create a single cell suspension. So you might have like a couple 100,000 single cells, and you squirt it on this slide, and you make sure that it's dilute enough that the cell-to-cell distance, which is all mathematically computable, is big enough so that when we park that droplet of acid, which is moving at 40 microns a second, when we park it over a cell, we get this burst of proteoforms that come. We would do a little extraction. And so here comes 20,000 proteoform ions. And because of this incredible sensitivity, we can now do this experiment. And we have some people, say from the Merkin lab, who's here today, Hadley, gonna help us with this. Yeah, so I could go on about this, but I won't. We can reconstruct the proteoforms that we see at the single cell level, and we're getting better at it, etc. So yeah, and we're sensing the beginnings of biological signals out of this. And so I come back, final sort of vignette here, single cell genomics. That is the ability to look at the DNA, or mainly the RNA. What is the program of transcription, from the genome to making RNA. That field's exploded. So RNA, single cell RNA-seq, here's their explosive curve of the number of cells possible from a single study, or from the field at large. This one's the field at large. So it got to 1 million cells pretty readily. And since that time is the way that the world is defining all the cell types in the human body. So we've disassembled 24 human tissues, organs, and we're massive single cell RNA-seq to the tune of about $2 billion. Chan Zuckerberg invested in the Human Cell Atlas, and a group at NIH called HubMap, or the Human Biomolecular Atlas program, combined and so this is reaching its sort of end stages. And so, for example, we know the kidney has, it looks like, about 80 cell types. And I go, yeah, cool, because now we need to know all the proteoforms in all those cell types. And that's the Human Proteoform Project. So there's a variety of reasons why I'm saying.
What is the market cost for this protein sequencing?
RNA sequencing?
Yeah.
The market size? So estimates differ, but it's,
Instrument post also.
yeah, they get you on the reagents, man. It's not the boxes. Everyone likes the shiny boxes, but they get you on the reagents. It's at least tenfold larger than the proteomics marketplace. So you're looking at a $50 billion, and that's sort of the mid estimate. So the estimate is that proteomics in the next, by 2030 will be a 38 billion, if you believe some economists, that the growth in protein level measurements, proteomics will start to approach something of that order. Yeah, so there's a large single cell experiments going on. We'd love to scale that. To do the analog of what single cell RNA-seq does, or the transcriptomics. They have a cell by gene atlas, they call it. And so here I'm coming with, now it's possible to do this, and we have a paper in review at Nature Biotechnology around that we're very proud about. But anyway, I've went on just long enough to bring me to the end. We're funded a lot by NIH. We have a National Proteomics Center, now housed within CLP, the Institute. It's about 60 people, and there's sort of 4 startups that have come outside or related to the lab. One of those is in the INVO Center, the incubation lab, the Q right over here. Yeah, I tried to catch that last wave. This was about $500 million of your taxpayer dollars at NIH. Yeah, I'll stop. I won't say much more about that. Wasn't able to convince them, but the ships are always leaving, so maybe we'll catch the next one. Thanks for your invitation. Thanks for the intro, Ed. That was great. Happy to stay and answer questions, and maybe there's a way to help us scale and bring some major resources and be the lighthouse for the world for the Human Proteoform Project. Thank you.
A few minutes for some questions.
Dave, I just had a question about the slide you had on the ovarian cancer cells.
Sure.
Can you speak up a little bit, Toby?
Yeah, I will try to. So you show that with the different decorations, then they're located spatially in different spots.
Right.
You also said that with the decoration, they can be in both benign as well as cancer cells. So my question is, how can you determine which cells are cancerous, right? If that decorated protein has function in both benign and cancerous cells.
Yeah, so they're welcome to molecular biology. The functional assignment is something that is a model created by people studying that protein, for example. The first thing I'll say is that the proteoform level knowledge is fundamental. If you want to understand the function of this protein, and you don't have that information versus you do, I mean, you have 2 very different views. The problem with biology is that it's fractal. This was Paul Allen's words to me. I would never forget him. He's like, yeah, you figure out something in biology, and then you look back and then there's another puzzle. And then you peel it and there's another puzzle. This is kind of like that, where certainly if you looked up in the protein databases, CRIP1, the protein, it would have a function, right? But that doesn't reflect our biology. You have to have proteoform level assertion of function, because it changes with these decorations. And the level, you can have moonlighting. So what does it mean to have a function for a protein? Well, it's not high enough resolution to answer the question. That's why creating a closed loop, if you will, for the proteome, that you had the players, and the ability to measure them. That's why it would be transformative, is because we're just low resolution molecular, like, oh yeah, it's the protein CRIP1. Great, okay, so we kind of know but meanwhile, it's got 2 very different functions in health and disease. Does that answer? But yeah, at least partially. Hadley.
So I would imagine when you have hundreds
of different versions of a protein, that the masses would start to overlap with the different post-translational modifications. Is there a way to extract that to know, is it a few methylations or is it an acetylation? That sort of thing with this method?
Yes, and if you can, this is the best slide I have at the moment. But yeah, you get a pattern of different masses and they can become overlapping at the MS, at what we call the intact mass level. And what you do is you go, oh, that's an 80 Dalton shift. Oh, that's probably a phosphorylation. Okay, cool. But if I had 6 phosphorylations, and they're all partially occupied and it does get complicated. That's why you need the woodshed. You've got to isolate, okay, that's a phosphorylation, great, let's isolate it, which we can do by spinning at a different frequency. So we isolate it, and then we chop it up, and we get all those blue flags, and we go, oh, yeah, it's phosphorylated, and it's at position 218. Or we see this for some proteins where, oh, actually that one phosphorylation at the intact level, it's actually 2 sites. 2 different positions in a 50-50 mixture of those 2. That's where, we deconvolute those things. It does get pretty hairy for some of the really complicated human proteins.
I'm wondering, can you, like, think about the woodshed, could that also be useful not just in proteoform, maybe looking at like some protein-protein interactions, like ones that might not even be, but like more transient interactions as well, or do they have to be covalently bound?
You can do something called native electrospray, where whole complexes can fly, whole biomolecular assemblies, and then you can controllably disassemble those. And then as you eject proteoforms out, like say you have a protein complex, it's got some RNA in there and it's got like 15 different proteins. Okay, great. So you biochemically isolate it, fly it, eject the 15 proteoforms. Oh, there's the proteoform landscape. Got it. Then I isolate and I take that out. It's a woodshed twice. You're chopping it up twice, but controllably. Totally possible to do. Yeah. Called native top-down MS. And it was first shown here with a Keck grant in 2012.
These abundances of different proteoform variants, I assume there's, abundance distribution in a single cell, there's abundance distributions in like an individual, there's also population level abundance distributions, right? And obviously you need like, very fine-grained tools to be able to look at the cell and like determine what, over what scale these abundance distributions are like interesting and relevant, but like, where do you expect them to? Interesting.
That was a beautiful breakdown of different a hierarchical breakdown of biological differences. Like you have population difference you need to know about. How much does proteoform variation in the you got to know that. That's not what the project is designed to answer. It'd be around about 500 or 1,000 human beings. But at a cellular level is where most of the action is in terms of, okay, how tight is the proteoform program of life? How much restriction do we have on that proteoform explosion? And a lot. And if I could tell you a story around ApoA1, it's a protein, it's the 11th most abundant protein in the blood. ApoA1, the protein, it has 18 proteoforms. When I look at your proteoform, ApoA1 and mine, we're both healthy, we don't have cardiac problems, they're gonna be really similar. So with that, proteoform program's conserved. And at the cellular level, it's the same sort of thing. It's not stochastic and all over the place. But that's why I think it's valuable to know the canonical healthy proteoform program. Because then you'll be able to see the tilt to disease. So it's not a quantitative answer, because it depends on some proteins have 3 proteoforms, some have 3,000. Yeah.
Do you understand why, you don't have exponential numbers of proteoforms? You're seeing a much smaller number. Do you understand the processes Or is it, they just can't exist based on some fundamental physics or chemistry? What's the?
That is a very timely question. And there's a startup called Nautilus Biotechnologies out in Palo Alto. And we should have this, my cohort, so we're both pulling for the Human Proteoform project. They have a single molecule proteoform detection technique. I have a technique that excels at down to 1% relative abundance. If you could go below or in the iceberg, how many below the surface, below 1% relative abundance are there? Answer: a lot. Our biochemistry is not perfect. It's it's amazing this whole thing's working. I mean, especially at 54 years old. Yeah, so that process goes wrong. And down there is it really important to get to 0.01% relative abundance? Because on tau, what the emerging story last year was that, yeah, we see 600 proteoforms, they see thousands. They're asserting that they do. Now they are a startup company. So, but I think that these two technologies are going to be complementary. One's truly a single molecule. They basically take a sample and they drop into a billion little landing pads, and then they probe it with 20, 50 antibodies. That's Nautilus. So I think it's a question of, you've got this canonical program. Say in aging, it starts to go awry in little ways. Your proteoform program, you start to lessen the control of that program. It's like, oh, more molecules are making errors in the reading, writing, and erasing of those marks. And that's aging. And so you should be able to measure that with this kind of noisy all the decorations that get imperfect, things get broken up, like in your eyeball. You don't renew the proteins in your eye as you age. And so what happens is those crystallins, the proteoform, it starts to become quite a mess as you get older. So that kind of, it's not like I've got the ground truth on how many proteoforms there are, but it's that proteoform program. Everybody loves to detract from my position about this because they just use the sea of oh, Neil, it's gonna take $80 billion, you're never gonna get there, right? No, there's a proteoform program that's running. We have to understand it. And yeah, there's the noise level below it.
What are the sort of mechanisms that would pull the 2 to the 40 possibilities to even thousands, or the relative abundances down there?
Oh yeah. So protein complexes are one such mechanism. Like yeah, I guess I do have one. Here's one with 9 phosphorylations that are possible, and 40 other PTMs. But here's what it is. There's 1, 2, 3, 4, 5, maybe 6 proteoforms. That's it. Above 1% and above. And so the mechanisms to control this are everything that evolved. You've got the kinase that's writing just to where it should, if you're healthy. And then you've got these erasers too that mop up. So the compensatory it's a beautiful program of life. I mean, it's
Not perfect.
no, definitely not perfect.
I think we are at time.
Okay, thanks so much.
Thank you, Kelleher.